Hi, this is Aaron Schwartz and David Perez, and today we're going to talk about a recent study about the anti-cancer properties of cruciferous vegetables, specifically chronic myelogenous leukemia, which is involved in the hyperproliferation of granulocytes, accounts for 20% of adult leukemias, and specifically is involved in a translocation between chromosomes 9 and 22. It results in this fusion gene, the BCR-ABL gene on chromosome 22, after this mutation called the BCR-ABL Philadelphia chromosome. It involves a translocation of exons between those two chromosomes, which results in that mutated tyrosine kinase protein. And so tyrosine kinases are enzymes involved in phosphorylation of cellular proteins, and specifically involved in cellular signaling downstream, which often leads to cell proliferation as well as other cell activities involving migration and differentiation. Now, the BCR-ABL protein is a mutated version of a endogenous tyrosine kinase c-ABL, and when this mutation occurs, you see oncogenic properties that lead to the constitutive expression of cell proliferation signals, which include ERK, RAC, J&K, and the STAT proteins, and thus we see this constitutive cell proliferation, which is so common to cancer. Right here we see a schematic, essentially, of where BCR-ABL, or once again, remember this is a tyrosine kinase, that is overacting, and we're basically creating these pathways to be constitutively activated, leading, as you see, all to proliferation and including decreased motility, which can contribute to development of tumors. Essentially, c-ABL is what's called a proto-oncogene because it's a gene that is very likely to be mutated into an oncogene which causes cancer. So as we've said, the abnormal activation of these protein tyrosine kinases are due to the enhanced expression of abnormal downstream signaling that leads to these cell proliferation or highly prolific cells that are resistant to apoptosis. Specifically, treatment is looking at how we can inhibit these enzymes, and much like we talked about in class, you look at the active site. So the binding pocket of ATP, since this enzyme requires ATP hydrolysis, is an essential target for chemotherapies. And here we see a simplified schematic of the structure, but we see ATP binding to BCR-ABL, which leads to its downstream effects causing chronic myelogenous leukemia, in opposition to current chemotherapies such as imatinib, which block this site. So once again, we're talking about treatments. We're going to try and target the ATP binding site, which will lower the tyrosine kinase activity and hopefully allow apoptosis to occur and remove the tumor. So here we have the chemical structures of various BCR-ABL protein kinase inhibitors, and as you can see, they're all relatively similar in structure. And we'll see that here, if we look at the protein structure in the active site of the enzyme, all of these molecules smoothly fit into the cleft that is formed that normally you would find ATP. And we see that these proteins have important effects on active catalytic loops and helices found in the active site. Unfortunately, these don't always work, and we see resistance due to specific point mutations in the BCR-able expressing cancer cells. Various other treatments are being looked at as to how we can treat drug-resistant cancer cells. One thing, funny enough, has been correlated to cruciferous vegetables. It's been shown that cruciferous vegetables inherently produce anti-cancer compounds known in a family called isothiocyanates. And recent experimental treatments with phenethyl isothiocyanate have been shown to have more effective treatment against resistant cell lines when in conjunction with currently used treatments. So here we see, again, more chemical structures of these isothiocyanates. On top we have an indole, which is also found in cruciferous vegetables. However, this study is specifically on phenethyl isothiocyanate. So this October, this treatment was developed and tested in vitro, so outside of the organism in a dish, in 2 constitutively CML oncogene expressing cell lines called K562 and Ku812, and various treatments with imatinib and phenethyl isothiocyanate at varying concentrations were developed and observed. We see a few techniques that were used, and we'll describe further when we look at the figures themselves. But essentially, we were looking at cell viability, protein content, that is BCR-able protein content, messenger RNA, as well as downstream effects of BCR-able and how well the treatment inhibits them. So here we're looking at the cell viability, specifically the metabolic activity of our cells treated with PEITC. And we're seeing that there is a steady decrease in mitochondrial activity with increased concentrations of imatinib and phenethyl isothiocyanate, indicating that these drugs are having an effect in these cancer cells. And hopefully, the goal, obviously, is to eradicate these. The MTT activity assay we went over in class, and this is simply an activity assay looking at the protein mitochondrial reductase, the enzyme and its activity on MTT in this transition. And by following that activity, we can determine the metabolic activity of those cells. Cell viability, we're looking at exactly what it sounds like, how viable this set of cells is after treatment. And what we wanted to see here is that phenethyl isothiocyanate has no significant effect on healthy lymphocytes, but significantly decreases the viability of our cancer-expressing lines. The tryptan blue exclusion method is simple but thorough in that viable cells will exclude this dye from their cytoplasm, thus remaining stainless, whereas non-viable cells take it up, and you can quantify viability based on that. Here we're looking at protein content of BCR-able in healthy lymphocytes to start with, and to be thorough to show that our cancer-expressing cell is in fact expressing the appropriate protein. And we're seeing that the higher concentration of phenethyl isothiocyanate, in addition to imatinib, we get a significant decrease in protein concentration, schematically shown down below. Now, our last slide was Western blot analysis, but here we see messenger RNA analysis via reverse transcription PCR. and we see that there is no significant decrease in messenger RNA content even after the same treatment that we saw on the previous slide. And so if we flip back and look at this, we see that there is a significant decrease in protein and no significant decrease in messenger RNA, indicating strongly that both of these drugs regulate at the protein, not the RNA level. Further, we are interested in the downstream effects of these chemicals. And BCR-ABL is a mutated tyrosine kinase that has enhanced kinase activity. Therefore, we're looking at phosphorylated downstream molecules of BCR-ABL, and we see that PEITC is having significant effects on decreasing phosphorylated forms of downstream molecules of BCR-ABL. Further, there are messengers downstream which also are phosphorylated and are directly involved in the signaling pathway that we showed earlier in the slideshow, showing that in healthy lymphocytes, they are much less expressed than in our cancer-expressing cells. Once again, we're showing that PEITC is significantly decreasing the presence of these signaling molecules, indicating that BCR-ABL, it has decreased function in these oncogenic cell lines. So, once again, these studies were all done in vitro, not in organisms. Thus, future goals are possible clinical trials and looking for the application of this molecule and perhaps other isothiocyanates in treatment of chronic myelogenous leukemia. Further, now that at least this study shows that phenol isothiocyanate has protein interaction and inhibition as opposed to messenger RNA, it opens the door as to determining a mechanism for this chemical's anti-cancer properties. In the end, as I pointed out on the slides, if you don't like vegetables, maybe this will convince you to start eating at least these types, because besides being concentrated and treated in these cells, there are additional studies that have linked cruciferous vegetables and many, many other types of vegetables and fruits to anti-cancer properties. Essentially, you just have to recognize that we are what we eat, and food is medicine. Here are our references, and we hope you guys enjoy.